breakthrough process to create cancer-killing drugs. A research team at Dartmouth College has developed a new strategy for drug discovery and development that can be used to produce targeted therapies against diseases such as cancer and neurodegeneration, according to a study published in Nature Communications. It is hoped that the process will also be useful in the large-scale production of new pharmaceuticals. The new technique uses a novel synthesis approach for a class of organic compounds known as tetracyclic terpenoids. Tetracyclic terpenoids are responsible for more than 100 FDA-approved drugs and are considered the most successful class of natural product-inspired pharmaceuticals. Until now, there was nothing like this available for drug discovery and development, said Glenn Micalizio, the New Hampshire professor of chemistry at Dartmouth. While additional development is expected to enhance the power of this new technology, I believe that we are at the beginning of establishing a truly enabling and potentially transformative technology for the pharmaceutical industry. The process combines two new chemical reactions that establish bonds between carbon atoms with a unique metal-centered ring-forming reaction also developed by Micalizio. The new technique allows for uniting molecular building blocks en route to a terpenoid skeleton in just a few chemical transformations. The result is a uniquely efficient and flexible means of enabling drug discovery in this area of natural product-focused medicinal science. Combined, these reactions allow for exploration of pharmaceutically privileged regions of chemical space through the straightforward conversion of inexpensive, commercially available chemicals into high-value, pharmaceutically relevant compounds. The research team's initial work has already led to discovery of what could be the most potent and selective modulator of the estrogen receptor beta, a nuclear hormone receptor that is of great interest in industry as a therapeutic target for a wide variety of illnesses. This is an important first step toward establishing a new technology platform to greatly facilitate drug discovery across a diverse landscape of therapeutic indications, said Micalizio. To demonstrate the value of the technique, the study describes the discovery of a molecule that is selectively toxic to glioblastoma, an aggressive and deadly brain tumor, while showing little effect on non-cancerous human neural stem cells and human astrocytes. Glioblastomas are incurable, and existing therapies have horrific side effects, said Artie Gower, an assistant professor of neurology at Dartmouth's Geisel School of Medicine. It is extremely exciting and encouraging to see that these novel compounds can selectively kill patient-derived brain tumor cells without harming cells from normal, healthy brain tissue. The team is currently conducting in vivo testing of the new therapeutic agent and further developing chemical aspects of the emerging technology platform. The work is part of an effort to explore and demonstrate the power of advances in organic chemistry to enable discovery of therapeutics for a wide range of indications, including neurodegeneration, neuroinflammation, and a wide variety of malignancies. Immunotherapy drug found safe in treating cancer patients with HIV. Study suggests the results of a study led by physicians at Fred Hutchinson Cancer Research Center showed that patients living with HIV and one of a variety of potentially deadly cancers could be safely treated with the immunotherapy drug pembrolizumab, also known by its brand name, Keytruda. During an ASCO presentation concurrent with release of a study in JAMA Oncology, Fred Hutch researcher and lead author Dr. Tom Aldrich said that in nearly all cases it was safe to use the drug in patients with cancer and HIV. The adverse events profile, a measure of the safety of the drug in the study, was not substantially different from prior studies that excluded such patients. The results, study authors said, are likely applicable to 5 similar drugs that block receptors known as PD-1 or PD-L1 on the surface T cells. Our conclusion is that anti-PD-1 therapy is appropriate for cancer patients with well-controlled HIV, and that patients with HIV and cancer can be treated with the drug and should be included in future immunotherapy studies, Aldrich said. The 30-patient trial studied only pembrolizumab, the anti-PD-1 therapy manufactured by Merck. Merck provided the study drug to the National Cancer Institute (NCI). The NCI sponsored the trial. HIV-positive patients with different cancers that might respond to the drug were included in the trial. Among the cancers treated were lung cancer, Kaposi sarcoma, or KS, non-Hodgkin lymphoma, liver cancer, anal cancer and advanced squamous cell skin cancer. Fred Hutch immunotherapy researcher Dr. Mac Cheever is director of the NCI-funded Cancer Immunotherapy Trials Network, which carried out the trial, and he is senior author of the JAMA Oncology paper. The study was conducted at 7 different cancer centers across the United States, including the HIV and AIDS malignancy branch of the National Cancer Institute, in Bethesda, Maryland. Overall, the safety profile of pembrolizumab in people with HIV and cancer was similar to that noted in clinical trials in the general population. Although the primary purpose of the study was to evaluate safety, it also provided a snapshot of the anti-cancer activity of the drug on these patients. One patient with lung cancer had a complete response to treatment, and activity was also noted in important HIV-associated cancers, including non-Hodgkin lymphoma, Kaposi sarcoma and liver cancer. An unexpected death on study from a rare KSHV-associated B-cell lymphoproliferation was noted in one patient, and while the association with therapy is still unclear, it has led to recommendations to use substantial caution if anti-PD-1 therapy is considered in the setting of KSHV-associated multicentric Castleman disease. The researchers concluded that anti-PD-1 therapy may be considered for FDA-approved indications in patients with HIV who are on antiretroviral therapy and have a CD4 count above a certain threshold, 100 cells per microliter of blood. However, more research is needed as to its effectiveness in the setting of HIV infection. The FDA, Friends of Cancer Research, and the American Society of Clinical Oncology have all recommended that HIV patients should be included in more clinical trials. The NCI has generally allowed patients with HIV to enroll on the immuno-oncology studies that it sponsors with PD-1 and PD-L1 inhibitors. However, this trial was one of only 2 trials sponsored by the NCI to focus exclusively on patients living with HIV, and it has been the first prospective trial to report its results. Exclusion of people with HIV in clinical trials is a long-standing problem that grew out of the poor outcomes of AIDS patients with cancer, before there were effective antiviral therapies for HIV, Uldrich said. In prior research, Uldrich surveyed 46 recent clinical trials that led to approval of cancer drugs, and found 30 contained explicit exclusions for patients with HIV, and 9 others where an exclusion was implied. This study was sponsored by the National Cancer Institute Cancer Therapy Evaluation Program (CTEP). Study drug was provided to the NCI by Merck Sharp and Dome Corp., a subsidiary of Merck & Co., Inc., via a collaborative research and development agreement. It was supported by U.S. federal funds from the National Cancer Institute (NCI), National Institutes of Health (NIH), under contract No. HHSN 26120080001E, NIH Intramural Research Program Support ZIA BC 011700 to Dr. Uldrich and ZIA BC 010885 to Dr. Robert Yarchone, and 1U01CA 154967 to Dr. Cheever for the Cancer Immunotherapy Trials Network. Diabetes drug alleviates anxiety in mice. The antidiabetic medication metformin reduces anxiety-like behaviors in male mice by increasing serotonin availability in the brain, according to a study published in J Neurosci. These findings could have implications for the treatment of patients with both metabolic and mental disorders. People with diabetes have an increased risk for mood disorders such as depression. Although the mechanisms underlying the relationship between insulin resistance, the precursor to diabetes, and depression are not known, studies suggest the neurotransmitter serotonin may be the culprit. In mice raised on a high-fat diet, Bruno Guiard and colleagues demonstrate that the insulin-sensitizing drug metformin reduces levels of amino acids that impair the entry of tryptophan in the brain and thereby limit its conversion into serotonin. The drug's antidepressant-like effects were accompanied by improved neurotransmission in the hippocampus. The researchers achieved similar effects by reducing the amount of so-called branched-chained amino acids in the diet. Bid to beat superbugs boosted by immune defense discovery. The fight against superbugs could be helped by the discovery of a potential therapy based on the body's natural immune defenses. Scientists have found that a molecule produced by the body, called LL37, changes the way cells behave when they are invaded by bacteria. The molecule acts like a fire alarm, experts say, warning the body's immune system of the infection and the need for urgent action. The team, from the University of Edinburgh Centre for Inflammation Research, focused on lung diseases caused by bacterial infections, which are a major cause of death worldwide. These infections are increasingly resistant to antibiotics, making them difficult to treat. Previous studies with mice found that LL37 instructs cells in an infected lung to call in specialized cells, known as neutrophils, which can destroy bacterial threats. In tests on human lung cells, researchers found that LL37 specifically targets infected cells, revealing the danger and producing a signal that summons the neutrophils. At the same time, this flood of LL37 into the infected cells causes them to self-destruct, removing the threat to other healthy cells before the bacteria can grow and spread. Experts say this discovery could lead to new approaches to treat these multi-drug resistant infections. Dr. Donald J. Davidson from the MRC Center for Inflammation Research said, "Our search for alternative and complementary treatments for antibiotic-resistant infections is becoming ever more urgent. Trying to boost the best of the human body's effective natural defenses, like this, may prove to be an important part of our future solutions." The study, published in the journal PLOS Pathogens, was funded by the Medical Research Council.